Welcome to the NOAA Science Seminar, Time-Space Portals, MatWorlds of Submerged Karst Sinkholes in the Great Lakes, which will be a presentation discussing 2 decades of exploration of Lake Huron sinkholes. This seminar series is co-hosted by NOAA Ocean Exploration and the NOAA Central Library. My name is Nina Pruzinsky, and I'm the Grant Science Support Coordinator working at NOAA Ocean Exploration in the Science and Technology Division and also with the Ocean Exploration Operative Institute. I am very excited to introduce you to our seminar speaker today, Dr.Bopi Biddanda. Dr. Biddanda is an aquatic microbial ecologist and carbon biochemist who currently teaches and studies the microbial biogeochemistry of the Great Lakes at Grand Valley State University. He received his PhD in ecology from the University of Georgia, where he explored new mechanisms of microbial carbon flux in the sea. His current research focuses on the development of real-time lake ecosystem chains observatories, the understanding of carbon-nutrient balance along land-lake gradients, and the exploration of submerged sinkholes in Lake Huron, the latter of which he will be discussing today. This project was supported via the NOAA Ocean Exploration Fiscal Year 2003 funding opportunity. This 2003 project spurred several other grant-funded projects supported by NSF, NASA, and NOAA's Thunder Bay National Marine Sanctuary, in addition to regular site monitoring funded and supported by Grand Valley State University, University of Michigan, University of Wisconsin, NOAA's Office of National Marine Sanctuaries, and Max Planck Institute of Marine Microbiology. Thank you very much, Dr. Biddanda, for your time and for choosing to share your exciting work with us today. I'll hand the floor over to you now.
Thank you, Nina. Hello, everyone. I'm very excited to share one of the most exciting projects I've been involved with in the last 20 years of my life. It has to do with exploring the submerged karst sinkholes in the Great Lakes as time and space portals. So let's get into this quote by Rachel Carson very early on. All of life on Earth has a touch of the sea in it. As you will see, even the ones in the bottom of of the Great Lakes, the largest body of fresh water on Earth, have signatures of the sea, especially in the shape of ancient marine salts that have been left over by the past seas here. They're still nourished by it. That's one example I'll show. The other is the beginnings. Yes, the beginnings of life are very hard to envision. But here we have two examples. One, on the picture on the right, you see of chemosynthetic microbes that are sulfur metabolizers in the bottom of the Great Lakes that are analogous to what you see in deep sea sulfur vents and seeps, where probably life first originated. And on the left two panels, you have these colorful cyanobacterial mats that too, they're recently discovered in the Great Lakes, that presumably were what the early Earth's past was like for perhaps the longest time, nearly over 2.5 billion years. It was a mat world in the shallow, sulfurous, oxygenless seas. So we'll get glimpses into this. And I want to note that this study was, supported by various agencies over the last 2 years. So, what do you do as an ecologist when you come across a whole new ecosystem, not just one species, but a whole new ecosystem or series of ecosystems? You begin with asking who is there, what are they doing, and the geochemist in you starts asking how fast are they doing it. But you will see that the really bigger questions, what is the meaning of systems, ecosystems like this to Earth's past, present, and maybe even the future? Those questions were initiated by the great colleagues and partners I had. So here they are, a wonderful uh, uh, group from the Thunder Bay National Marine Sanctuary dive team and NOAA's ROV teams, and amazing colleagues from NOAA and other universities that made this possible. So I also want to tell you to just sit back and enjoy a picture show. This is going to be a picture story. So I'm going to leave behind the hardcore journal, peer reviewed data behind, and just tell you a picture story, and then we'll pause to take questions. So the very first grant was from NOAA Exploration in 2003, and the very current one that we have going on is from NSF Oceanography. And in between, we have about 30 journal articles, 2 Nature publications. We made 2 journal covers, Ecosystems and the Great Lakes, a Journal of Great Lakes Research, and we published 2 feature articles in EOS, the newsletter of American Geophysical Union. So let's get to see what I will cover in this today's seminar. What is the microbial ecology of Lake Huron sinkholes? Do modern-day microbial mat ecosystems resemble life in the shallow, anoxic, and sulfurous seas of the early Earth? Could mat worlds like these have played a role in oxygenating our planet over geologic time? Could active vertical migration, daily vertical migration of microbes in the mat have anything to do with this, meaning oxygenation and carbon burial? Can life in submerged sinkholes serve as models in our ongoing search for life elsewhere? So here's an image of what astronauts on Apollo 11 saw in the 60s from looking over from the surface of the rocky moon. And amongst the many poignant thoughts they must have had is also, oh, we missed the chance to name our planet correctly. Should have been ocean or should have been water, isn't it? Yeah, water is everywhere on Earth, and microbes rule there and everywhere else, as you will see. I will start this off with the cocktail napkin, probably a misquote, so bear with me. So in wine there is freedom, in wine there's wisdom, beer there is freedom, and water there is bacteria. The last is definitely true. Here's on the right, you have an epifluorescence microscope image of a milliliter of water filtered onto a filter paper, and using fluorescent stains, you can bring them back to life. So they glitter because the DNA gets stained. So there's nearly 500,000 or 2 million cells in every milliliter of water on the surface of the planet, the oceans, freshwater streams, wetlands, and so on. So that adds up to 1 to the power of 10 to the 29 living microbes. That is huge. And that is actually more than all the stars there are in the known universe. And we haven't even counted what's in the soil, sediments, and other life, like including microbiomes within us and other organisms. So talking about water, the Laurentian Great Lakes of North America compose the largest contiguous body of fresh water on Earth, nearly 1/5 of liquid surface water on the planet. So the area we will cover today is marked by that red spot in the middle. You want to remember this area. It's in northwestern Lake Huron. Much of the seminar will explore submerged sinkholes here. This is a geological map, and you will see that the lower Great Lakes, Michigan, Huron, Erie, and Ontario, are all underlain by sedimentary bedrock, mostly composed of limestone or dolomite, okay, carbonate rocks. And there also these were sedimented in the Paleozoic, 400 million years ago or so, and the ancient seas left their salts behind, especially chlorides and sulfates. And today's aquifers wash through these and carry these salts, and in long conduit time makes them lose all their oxygen when they come out. Little more geological background. This study, you can see this area of northwestern Lake Huron and adjoining continent of Michigan, the southern peninsula, are very complex, and they've got this concave structure of layering of different ages of rocks. Sedimentary rocks that are then in the bottom. There's impervious rocks, and the sedimentary rocks are limestone and dolomite and some gypsum. So you see carbonates and sulfates. So you can expect them in the groundwater in these zones. How do sinkholes form? They form all the time on land, and they become news. When that happens in the lake floor, That's often not doesn't make the news. But the way it happens is rainwater percolates through limestone bedrock, making caverns and caves, and water actually can flow uphill if it is blocked by impervious layer, and then they can erode limestone in the bottom of the Great Lakes, for example, and then they cause sinkholes over time. And emerging groundwater is sulfur-rich and oxygen-poor, mimicking early Earth's shallow seas. So now we're zooming in to northwestern Lake Huron, and this is the city of Alpena for reference. And there are many areas of onshore karst sinkholes and offshore karst sinkholes. Okay, and onshore sinkholes can be seen from a flight in an aircraft. They form nearly perfectly circular holes in the ground. And nearshore ones, which are full of springs and large crater-like sinkholes, can be seen very shallow ones. You can even wade into and see gurgling sulfurous streams come out. And offshore, this is one of those sinkholes we're going to look closely at. This is the Middle Island sinkhole. It's about 23 meters deep, and about 5 to 10% of ambient surface light gets down. So it can potentially support photosynthetic life down below. And offshore, this was an accidental side scan discovery. You can find aphotic sinkholes where no light gets down. And so we'll look at closely at these two systems, the sunlit middle island sinkhole and the aphotic isolated sinkhole. So you want to, these isobaths are at 1 meter intervals. So, the closest, the number one is about 25 meters and number two, the offshore sinkhole is at about 100 meters deep. So coming to the shallow sunlit sinkhole, we identified two regions. This is a multi-beam high-resolution map, so you can see cracks here and there, and you can see like a crater-like area where the limestone has just dissolved, and you have an open area here. So this is one point where groundwater fills up, which we call alcove, and then overflows over a sill. Remember, this is 23 meters deep. It overflows over a sill that's about 10 meters deep and flows in an underwater water flow. Okay, so remember the groundwater is cold and it's dense because of the saltiness. So it actually hugs the lake floor and eventually gradually, like almost like a chromatograph, it dissipates down across into the bigger lake. So as it flows down from this alcove into the arena, down this underwater waterfall, you can see there's a big thermal and density discontinuity. You can see almost see that layer on the divers down here. And down below, you see amazing, brilliant purple mats, cyanobacterial mats, being nourished by this groundwater full of sulfur and no oxygen. A little bit more about what are the production mechanisms in these sinkhole ecosystems. You can see overlying Lake Huron oxygenated water and underlying anoxic sulfate-rich groundwater. And a little breakdown. The regular algae flourish up there in the lake. Then you see some of these chemosynthetic microorganisms, which are white in color because they don't have any pigments. Then there are photosynthetic purple cyanobacterial pigments. A little bit more about the community composition here. You will see that these mat communities are taxonomically and functionally very diverse and versatile. So one thing here is that the cyanobacteria, mats here, are capable of two types of photosynthesis. The ancient anoxygenic photosynthesis, which doesn't result in the production of oxygen, but involves the metabolism of sulfur in sunlight, and the modern oxygenic photosynthesis, which results in the production of oxygen. So you can see even in photosynthesis, they have both ways of doing it, versatile. And bacteria targeted gene analysis clearly shows that it's dominated by bacteria, dominated by cyanobacteria. These are the first of plants to start making oxygen on the planet. Okay, and other diverse heterotrophic and other bacteria. And whereas looking at the chemosynthetic mats, the void sulfur oxidizing bacteria and archaea, you see that through genomic analysis, the red and blue show you that there are many different sulfur oxidizers, and some of them have genes that are verified to oxidize sulfur, oxidizing using hydrogen sulfide to actually synthesize organic matter, like the ones around the thermal vents and the sulfur seeps in the sea even do today. So comparing the composition of lake water, Lake Huron water to venting groundwater, you'll see venting groundwater is salty many times, has got high levels of chloride, and has a constant 9 degrees Celsius temperature, okay, which is probably the average millennial temperature of the air over 45 degrees north latitude in Michigan, around this area. The lake temperature varies depending on the season. It can be warmer or colder. pH levels you'll see is almost 1 unit lower, probably because all the oxygen is gone in that. Meaning, and also you can see in terms of organic matter, much of the organic matter, or dissolved organic carbon, has been respired away, resulting in inorganic carbon, meaning there's more carbon dioxide dissolved, and probably the reason why pH is so low. So all the oxygen is gone, it's high in sulfate, and these are conditions that prevailed in early shallow seas of the Earth in the early days. So another thing is groundwater emanating at different locations in the Lake Huron have somewhat similar signature, although some that are deep sinkholes have been found to have higher chloride and higher sulfate. But it suggests they're all coming generally from a common aquifer source. So looking at the profile here, these are beautiful classic textbook examples of a double thermocline. You see here where this is the transition zone where lake water meets groundwater. You'll see, temperature drops drastically within just within less than a meter, and then dissolved dissolved oxygen goes to nothing from well oxygenated condition. pH drops by 1 unit. Specific conductivity jumps up by almost 1 order of magnitude. Who are these microbes? When you look under the microscope, you see these brilliant purple filaments. Those are cyanobacteria. And you see these dark or colorless filaments. They are the chemosynthetic archaea and bacteria, sulfur oxidizing ones. And you will see that both of them are motile. It's part of their mat building philosophy, I guess. So if you take a cross section that goes through this surface surface layer of cyanobacteria down to chemosynthetic sulfur oxidizing bacteria, sulfate reducing bacteria, and at the very bottom you see carbon rich methanogens, okay, where methanogenesis or methane origin happens. These are very closely interconnected layers of organisms, a near perfect redox tower. So the waste of the one is the resource for another. So very closely knit consortium of organisms that makes this mat ecosystem work. So we did some experiments in the lab to look at how motile these organisms are. You disperse them in a petri dish, and you put a aluminum foil over it and shine light, and they will come to the light. So they're phototactic. Also, put pebbles or shells on top of an intact mat sediment core, and within several hours, those pebbles or shell pieces disappear, presumably because cyanobacteria are climbing over them to harvest light, to seek light, and in the process they end up subducting them down below. And these issues are very important. They help with the carbon balance in this ecosystem and resulting in oxygen release. We did several in lab core intact core experiments and in the field benthic chamber experiments, and we find nearly all of the oxygen that is produced is consumed with a slight leftover, meaning not much direct carbon synthesis is left over from day to day, but a little bit is, especially on longer day cycles. But that's what little bit could add to a lot over 3 billion years of a mat world biosphere, if that's how the biosphere was in early Earth. The little can go a long time. Underneath underneath the mats, there is a huge amount of carbon, very carbon-rich sediment. So sub-bottom profiling has revealed that there is, for example, in Middle Island Sinkhole, this is lake surface, sediment surface, there is about 18 meters, 17, 18 meters of sediments. Okay. And they're very carbon rich compared to lake sediment, typical lake sediment, which is 5 to 10% carbon. And lead and cesium dating suggests that their age is about 7 to 10 thousand years. Means they are, since deglaciation. But this suggests that there's a 1 to 2 meter accumulation of sediment per every 1,000 years, every millennia. That is absolutely the highest rate measured anywhere, if this is correct. We also put sediment traps, in this location in the middle of the theater, and analyze them for stable isotope composition, both carbon and nitrogen. Also analyze the plankton and the organic matter in the sediment. The hypothesis was that annual layers of mats would get buried in, and that's what's making all this carbon-rich sediment. But no, the sediment below doesn't have any mat signal. It is full of the planktonic signal. So it means the mats are burying planktonic carbon, and so as carbon gets buried, oxygen is potentially released. We did many experiments to track the diel vertical movements of these organisms using a GoPro camera that was set up in the sinkhole bottom. In this video, you can see the location and the arrangement of this time-lapse camera. And you see there's various patches where the mats have lifted naturally and gone away. We believe that's the way mats may disperse to other isolated locations, sinkholes. This is just a year-by-year overview of how during the day and during the night the mat looks. During the day they are bright purple, and during the night they are more whitish. So the inference is that likely that the photosynthetic cyanobacteria are coming up during the day, and chemosynthetic sulfur oxidizing bacteria are coming up to harvest the hydrogen sulfide at night. And so these are just 1 to 2 millimeter journeys, but they could have been the largest daily synchronized mass movement of life on early Earth. And this is just a schematic of how that would happen during the day. The cyanobacteria migrate up, and they reverse at night, and the opposite happens during the night. So are they optimizing the use of light and chemicals? So this is a half-hourly time lapse over a day from midday to the next midday. And you see a wobbling meter scale there. That is a tilt meter that's looking at the flow of groundwater. So we did microprofiling at every 100 micrometers using hair-thin probes for oxygen and hydrogen sulfide in intact cores that were in the lab. under simulated temperature and light conditions. And you see during the day the oxygen levels increase, and at night they drop. And hydrogen sulfide concentrations increase during the day, and they drop down during they increase during the night and drop down during the night. So this mechanistically explains the migration of photosynthetic organisms to harvest sunlight and make oxygen during the day, and of the chemosynthetic ones to come up and harvest hydrogen sulfide at night. And you can see they release oxygen on longer days and late afternoons. So here's a simulation of that from night. The moon depicts night half hourly, and during the day the oxygen increases and hydrogen sulfide draws down. So why do they move, and why should it matter? In daylight, purple photosynthetic filaments move upward seeking sunlight. Net photosynthesis resulting in the release of oxygen from the mats occurs in the late afternoon, longer days. When oxygenic photosynthesis is released from sulfide inhibition, photosystem 2 is inhibited by high levels of sulfide due to sulfide consumption by anoxygenic photosynthesis and chemosynthesis during the day. At night, chemosynthetic white filaments move upwards to scavenge remaining oxygen above while using hydrogen sulfide generated below, following the daily vertical change in the oxygen hydrogen sulfide interface, which moves up and down across those just a few millimeters. Such diel vertical movements may have been the largest daily mass movement of life for billions of years and favored mat building, sequestered carbon, and released oxygen. Today, the largest mass movement of life daily is of zooplankton doing diurnal vertical migration in the oceans and in the lakes. But it might have been this sub-millimeter journey might have been the largest daily movement of life for 2 to nearly 3 billion years. And over time, it might have really mattered. So we are off to the deep sinkhole as a model of marine seeps and vents. This is a 100-meter deep ecosystem where no light penetrates in Lake Huron, and uh, this is a sub-bottom side sonar image, and this is the ROVs we use to sample that. We actually profile the water column using a CTD, a conductivity depth temperature sensor attached to the ROV, and you can see temperature, pretty uniform in the deep. This is very close to the bottom, just the bottom 10 meters or so. It's about 4 degrees Celsius Celsius in the Great Lakes at that depth, and then you can see it warms up to about 8 or 9 degrees, which is the temperature of groundwater. And conductivity increases by nearly an order of magnitude, okay, meaning it's salty. Groundwater is salty in that interface. Also, the groundwater being warmer is relatively lighter, but it's denser because it's saltier. So after it comes out, it layers out like an umbrella here, like a nephloid layer. And using a peristaltic pump and a strong peristaltic tubing and pump, we brought up these samples at these discrete depths, and you can see the nephloid layer samples are dark like this. And this is I'll get to that in a second. Here you can see a profiling we did just over 1 meter above the bottom, and you can see the conductivity hotspots here. High conductivity hotspots also coincide with the high temperature of the groundwater signal. So they go hand in hand. High conductivity, more salty, sulfate and chloride rich water, along with the warm groundwater seepage. So in this nephaloid layer, we made some of the very first chemosynthesis measurements for a Great Lakes water column. Um, and you can see these are similar to images that the submersibles like the Alvin sees in the around deep sea vents and other even cold sulfur seeps in the marine environment. So similar to those found in marine seeps and vents. And the organisms we've isolated from here, Biddanda and other chemosynthetic organisms, are also of the same genus, at least, of the ones found in marine systems. So, what are some of the recent discoveries in this thing? Are these restricted just to Lake Huron, or are they elsewhere? Anecdotal stories from sports divers and all. We have reports of them across all of the lower Great Lakes, but scientifically we have been able to map in 2018 through 2020 using multi-beam mapping about 2 dozen new sinkholes. Some of them are twin holes, and some of them they're already at 120-meter isobath, and they plunge down about 18 meters further. So, and there are indications they have groundwater just from the sound wave contrast, but we are yet to sample and study them carefully. In the Lake Erie Basin, we recently explored what's called the Great Sulphur Springs, and also see cyanobacterial mats down below. And uh, the National Marine Sanctuary in Lake Michigan and their colleagues at NOAA have done sub-bottom surveys and see clear signals of a community of sinkholes in western Lake Michigan shores. So we believe they're all over the Great Lakes, and maybe abundant because they leave no surface signature, so they are very hard to find unless you really run into them or purposely looking for them. So where else on Earth are such microbial mat communities found? So this is the Middle Island sinkhole. And you see, if you're wondering what are those projections, they are half to 2 feet high mounds of mats. Okay, they late in the summer, the peak of their growing season, there's lots of sedimentary gases, especially methane, that builds up and lifts up this carpet, delicate mats into carpets, and then they actually tear up sometimes and float away with the bubbles. So maybe that's one of their dispersal mechanisms. So the question of where else? Yeah, in the dry valley lakes of Antarctica, the permanently ice-covered lakes have the same communities, okay, similar communities, and as you can see here. And how do they get from one place to another? Are they all hiding out in refugia that are low in oxygen and high in sulfur? Those, are they all around the planet in hiding? Those are the bigger questions. They're in other lakes too. This is an example from Lake Cadagno, which is a sulfur spring-fed lake that made the cover of Science a while ago. So where not on Earth, where else could we find such mats? As we know, there's the many water-containing bodies, even on our solar system, extra-terrestrial. There's at least 9 bodies of water other than Earth that contain significant amount of water. Europa and Titan and those come into mind. For example, Europa has more water in its oceans than all of the water on Earth. In fact, often it sends plumes of steam, water steam, out of its icy shell. Okay, what might we find if we probe below the icy cover? Okay. So we propose that something like the Middle Island sinkhole, the euphotic one, or the sunlit sinkhole, which is only 25 meters deep, could be a working model during, with ice or without ice, for NASA to, practice collecting, possible samples in faraway places like Europa. So Earth microbial mats can not only provide a window into past, maybe they also can provide a model for life in extraterrestrial hydrospheres. What might we find? So here's a timeline of life. I'm coming to the conclusion here. Time of photosynthesis and oxygenation in Earth's atmosphere and the oceans. For the longest time, we had very little oxygen, and even then, it was less than 1% of what today is. This is a log scale, so there's a big changes now. It's only recently that we had this huge spike, which is 23% of the atmosphere, which is 100% of depicted here. In between, we had the ancient first photosynthetic process and oxygenic photosynthesis. Then we had a mixed anoxygenic and oxygenic photosynthesis going on for nearly all of the Proterozoic, which is from like 3 to 0.5 billion years ago, until the emergence of larger eukaryotic and, animals and life on land and so on. So presumably, it was a mat world biosphere, mats clinging to the deep sea vents and then to the shallow as a thin biofilm into the shallow shelves of the sulfurous, oxygenless seas. A Proterozoic mat world may have primed the biosphere for peak oxygenation and carbon burial in the last 500 million years. That's the question. So to wrap all this up, time, water, geological forces have somehow converged to create underwater sinkholes where oxygen-poor and sulfur-rich groundwater support prolific microbial mats resembling life on early Earth, and they contribute to today's biodiversity on Earth and are, I would argue, worth conserving. Photosynthetic cyanobacteria in shallow sunlit sinkholes may be the modern-day analogs of the Proterozoic, whereas chemosynthetic mats in the deep aphotic sinkholes are analogs of deep-sea vents and seeps. Motile mat worlds like these may have been critical for surviving the turbulent, shallow Precambrian seas and eventually oxygenating our planet. And modern-day mats, microbial mats, may be also working analogs in our search for life elsewhere. We're not done. There's still several remaining emerging questions that pop into mind. What is the abundance and distribution of sinkholes in the Great Lakes? What is the subsurface plumbing in the Great Lakes Basin really like? The largest body of fresh water. I mean, just by the resource value of itself, it merits study. And what is the role of sinkholes in it? Why have we mapped so little of the Great Lakes floor compared to even our coastal seas? What is the composition, quantity, and age of groundwater venting at different sinkholes? We have no idea of some of these things, basic things. Do only sinkholes with active groundwater venting that have no oxygen and high sulfates nurtures microbial sinkholes, microbial mats, whereas others don't? How do mat microbes disperse and colonize very distant and isolated sinkholes, in the Great Lakes Basin and across the planet? How sensitive are sinkhole mats ecosystems to changes on land, say land use change, precipitation, warming, etc.? What is the contribution of sinkhole microbial diversity to Earth's extent, modern-day biodiversity and physiological potential? Remember, these microbial mats are very taxonomically diverse and functionally versatile. Potentially, they can do everything in a model redox tower. What are the biochemical and pharmacological potential of extant mat ecosystems? Should we conserve and protect Earth's remaining mat worlds? I will end with this thought, which is obvious. we pick something like a microbial mat. Maybe even a one or two microbes out of it, or even a piece of mat, or a whole ecosystem. We clearly see it is linked to everything else. I'll lead you to this further reading if you're interested. This one just came out in EOS as a feature article in the January issue. EOS is published by the American American Geophysical Union, and in under eos.org you can look for microbial mats or sinkholes, and it'll pop out. And I'd like to end with a thanks, and I will be happy to take any questions. Thank you.
Thank you so much, Dr. Biddanda, for your presentation. This is Lisa from NOAA Library, and audience, we have lots of time to answer your questions, and we'd love to hear them. So please look to your right and type them in the Q&A chat, and I will upload them for Dr. Biddanda. But Nina, did you want to start us off with your question?
Yes. Dr. Biddanda, I was wondering if you could provide some advice to students and other early career researchers that want to pursue this field, and just provide some advice on how to get started and get more involved in this research. And feel free to discuss how you got into this field as well.
Well, this project has been like a godsend for involving students. So most of the students involved in my program here and that of my colleagues have in-person experience with this, snorkeling and diving and collecting samples and working through the other inquiries and so on. So it is a magic project for us. It's just designed for such inquiries. Yeah. Uh, Advice, to stay curious, right? There are so many questions beyond what is there, who are there, what are they doing, how fast they're doing. There are these views you can take that are broad earth scale things. You can begin to wonder, what is the relevance of one small thing to the big picture. uh, I think they're in some obviously some of the biggest challenges for us, even with issues like dealing with biodiversity conservation and climate change and other matters. So I would say, um, be interested in the small, but dare to think big, and try to see the big ecosystem or the global picture in everything you do. So um, that'd be the kind of broad scale um, um, idea that I would answer that question with.
Thank you very much, Dr. Biddanda. We did get a question, which I will put across the screen now. And this person asks, you mentioned that there is a change in the sinkhole mat world during the day-night shifts. Have you observed shifts throughout the year, such as during the seasons?
Yeah, very good question. All questions are good, of course, especially in these systems where we have studied so little. Yeah, we definitely seen them across the ice-free season, meaning from May through September. But we don't know what happens during the winter in the dark. We've never dared go through the risk of diving or even sending ROVs at that time. Uh, I would speculate something similar happens. These are synchronized daily movements to light, but maybe they move little during the winter months. But I think they maybe they amplify during the summer. In the spring there's it's less movement, in summer there's maximum, in the fall less, and in the winter very less. But of course these are all very hypothetical. From what we have seen in the growing season is that they actively migrate, dramatically changing the color of the ecosystem from purple, brilliant purple during the day, to bland white at night. So this is in their circadian rhythm. So I would assume they would do it. Or they would do it around the year, based on sunlight and so on, because they are responding to the chemical gradient that's moving up and down. And the chemical gradient is also partly driven by light and photosynthesis and the generation of oxygen, small amounts of oxygen at the surface of the mats. So that's the long answer to that. Very, very good question. Thank you.
Thank you. Here's another question we just got. This person asks, has there been any microbes that have been uncovered from these fresh underwater sinkholes that could be of harm or helpful to our current ecosystem?
As best as I know, these are very pristine natural ecosystems, and we don't know of any that at least as of now that would harm us or other ecosystems. Obviously, they are naturally spreading everywhere. Otherwise, they wouldn't be in distant places at the current time. So I would presume they are natural ecosystems, and they are harmless, but we don't know for sure. On the other hand, I would propose that, because of the intimate nature, everything happens in sub-millimeters and micrometer, interactions here. I would I would assume they and presumably they have a very long history, easily thousands and thousands of years, if not millions. So they would have some of the most well-developed, you know, biological warfare kind of thing, where you have elements of cooperation, competition, and all those things worked out, predation. So in this case, viral lysis, for example, of cyanobacteria would have worked out. So they would be very rich in biochemical compounds that may be of a great pharmaceutical value for us. So those potentials definitely exist. And one thing that I should point out is that also that something I did not really emphasize on is the amount of carbon per unit area these mats are able to do. They know about burying organic carbon. If we could harness some of their capabilities, that might be really helpful in burying, addressing part of our excess carbon dioxide problem. They know, there may be small amounts of them in small areas in refugia, but they know how to bury carbon, and they bury planktonic carbon, which is exactly what we want. We want planktonic carbon buried away so that they can be helpful in removing surface carbon, which is coming from the atmosphere. So there's many potentials, I think. Understanding life in these sinkhole microbial mats that can be helpful not only just in biodiversity conservation, but also in human and ecosystem welfare. Yeah, some of this is obviously pure speculation because we haven't done the work. Thank you.
Thank you. That's really interesting. This next question is anonymous, but it asks, what did you think was the most exciting thing that you discovered during this research?
Oh, there's so many because so much of this is new. One of our University of Michigan colleagues discovered what is called what is the gene signals, not the organism itself, but the gene signal in the isolated sinkhole, which is the deep water sinkhole, the aphotic sinkhole, which only had the white mats that mimic the deep sea or marine seeps and vents. In a ROV scoop of the sediment and mat complex, they discovered genes of an organism called um, Thiomargarita. I forget the last this thing. It's a sulfur oxidizing bacteria. It is the largest bacteria that we know. It is millimeters in size. It's visible to the naked eye. Okay, the name is Thiomargarita namibiensis. Okay, found in the deep sea sulfur vents. Okay, and we haven't found the bacteria itself under the microscope, but just to realize, wow, there is marine analogs right here in our backyard, and not thousands of meters deep, just 100 meters deep. That was amazing.
Thank you. Another question asks, how many species of microbes have you found?
I'm not on top of it right now. It's definitely hundreds, definitely hundreds, and the peer-reviewed publications from my colleagues have published metagenome and other analysis of this. Not only just not about what genes are there, but what genes are being expressed on things like transcriptome, the genes that are actually active. And so they've been discovering more and more, species as well as functional capabilities. So that field is growing and is ripe for, further work.
Excellent. It looks like that might be your last question. So I'm going to transfer the screen to Nina. Thank you so much for this presentation.
Thank you.
Yes, thank you for sharing your research with us. And I also want to thank Lisa for facilitating. I wanted to let everyone know that the next NOAA Ocean Exploration Seminar that is co-hosted with the NOAA Central Library will be on August 21st at 3 p.m. Eastern Time. And that will be with Dr. Amy Gussick, who will discuss her work on the paleo landscapes, paleoecology, and cultural heritage now submerged on the Southern California continental shelf. So make sure to mark your calendars, and more information will be coming out shortly. Thank you everyone again for your time today.